All right. Hi, everyone. Welcome to our Google Plus Microbiology Journal Club. And after some technical difficulties, we finally got it online. Please, Google, a note from us, stop changing the way that the chats are starting. It makes things very confusing. We would very much appreciate that things are kept consistently so we could get these things going at a reasonable time.
I second that.
So today we are reading a paper that came out of San Diego State University, and it's called Subdiffusive Motion of Bacteriophage in Mucosal Surfaces Increases the Frequency of Bacterial Encounters. And so I'm not as good as Laura because previously I think she read off the screen once to the actual title. But
that was really hard too. I started doing it and I was like, oh, this is a mistake. I don't know.
Would be easier if I could see myself bigger, but I'm in a tiny screen at the bottom. But at any rate, this paper, which is really interesting, has drawn a lot of media attention actually, because it talks about the hunting behavior of phages in mucus. And so before we get too far, I will just let Laura introduce herself and stuff that she works on.
Sure. So my name is Laura Williams. I'm an assistant professor at Providence College, which is a primarily undergraduate institution in Providence, Rhode Island. And right now, I'm kind of, staggering towards the end of the semester. But one of the things that I do here is I have a research lab and we work on predatory bacteria. And so this is particularly interesting to me because I think there's a lot of parallels between bacteriophage and predatory bacteria. So it's interesting to see this, especially when they start discussing phage therapy and applications towards phage therapy. That's something we're certainly interested in. And so I'm looking forward to talking about this because otherwise I might not have read this paper, even though it looked really interesting. It would have been hard to find time to do it. So I'm really glad we chose this paper.
Well, and that's part of the joy of Journal Club, which is that, when we're here, we get to talk about all these different papers that we would never read otherwise.
It's true.
It's so my name is, so I haven't introduced myself. My name is Karen. I'm a PhD student at Simon Fraser University, and I study the potential of bacteria called Francisella tularensis. And I look for the virulence, the proteins that contribute to its virulence, in particular attachment, invasion, and replication. And so this paper is interesting to me from two levels. One, it's because I actually get to read a paper about invading microbes and attaching microbes from a very different perspective from all of the different symbiosis papers and microbiome papers that we've been reading in the past. So this is, and secondly, I was really interested in this paper because it uses mathematical concepts and applies them to behavior of microbes. And so that's something that a direction that I'm currently interested in looking, because I find that there are a lot of behaviors in biology that we can't detect, or that it's very hard for us to understand how things actually work in biology because we're limited by the techniques that we're using. And so I really enjoy when people use mathematical concepts because it does simplify the concept a bit, but then in the end it's like, oh, that is something, a way that you can apply this method and you can learn something from it. And that's something I really appreciate.
Yeah, I agree. I think it provides some interesting kind of methodological and analysis innovations to a topic that certainly has a lot of relevance. So I think that was well described. Before we get too much farther too, I want to give a shout out to our missing microbial musketeer, Mark Martin. Oh wow, that's some alliteration right there.
Yes.
Missing microbial musketeer, Mark Martin. Wow. So he is, not able to join us today. I think this semester is taking its toll on quite a few people in terms of the workload. So we're looking forward to having him back when he's able to join us again. And if anyone else wants to join us, we are more than happy to have other people join the discussion too. So I thought I would put that out there before we got started.
Yes, so because the nice thing about having, at least for me, because I come from a very small institution at Simon Fraser, we have a very small microbiology department. Most of us, there are a lot of people that work on viruses, and I think the number of people that work on bacteria, or the number of labs that work on bacteria, I can count on one hand.
Oh, wow.
So it's very nice to have, it's really great to have like a journal club that I can come to where we discuss these recent papers and recent findings with other microbiologists in the field. And so it's nice to meet everyone, and we really encourage you to come out. All right.
Yeah.
So let's get right into the paper. I've been talking a lot, so Laura, why don't I let you summarize the?
Oh no, that's okay. Yeah, okay. I mean, I would just just very broadly, I think one of the really neat things that this prompted me to think about. So first of all, I'll provide some context to say that I'm currently teaching a non-majors class on microbiology. And they took an exam today that covered kind of the microbial diseases portion of the course. So they had to think about viruses and their differences from bacteria and the differences from microbial eukaryotes that cause disease. So I'd been thinking about things like adhesion, I'd been thinking about things like bacterial motility, and it was really neat to read this. And the writing is very very accessible in terms of getting people on the same page to understand when you're talking about a bacteriophage, that bacteriophage is not actively moving in any way. So viral particles, there's this, the argument that they're not actually living because they can't replicate on their own, they have to hijack a host cell. And in concert with that, you get the idea that these bacteriophage are not in fact hunting and destroying the same way we think of like lions hunting things, or even in my research, predatory bacteria, which are moving around and trying to actively find their prey. So for bacteriophage, this paper is really interesting because what it kind of starts to get at the different ways that bacteriophage can, I mean, it's even difficult to say it like this because they're not actually doing anything. It's just there's been selective pressure for this interaction between the bacteriophage and the mucus layer that allows them to kind of define their search space. So I think it's neat to think about this because even as I'm talking about this, I'm using some language that doesn't capture, in fact, what's actually going on, because the bacteriophage are not actively doing anything, which I think is an important point to kind of sit back and be like, oh yeah, that's true. They're not really doing anything, but there's this interaction that ends up resulting in kind of a more defined search space than you otherwise see due to like random motion.
So you bring up an interesting point, which was I really wanted to hear your perspective in terms of, because you studied predatory bacteria. And so you've mentioned that predatory bacteria have active ways of locating their prey. And so is that by molecule, or do they secrete some kind of molecule, and then or are they detecting prey via a signal that's being diffused in space? Or
yeah, so there's a paper that just came out in PNAS that I'm working my way through that's talking about different cues that predatory bacteria might use. So the most well-studied predatory bacteria, Vibrio bacteriophorus, has flagella. So they're in fact very, very fast swimmers, if you want to think about it that way. So when my research students look at it under the microscope, you can see in the field of vision prey cells like E. coli or Pseudomonas, and then this tiny cell will just go pew, pew, across the field of vision. And they're very fast, and there is some indication that they are potentially, there are people who are looking at chemotaxis to try to understand, are they following a concentration gradient of a particular compound that prey cells are producing, or is it just or is it primarily has to do with accidental collisions? And I think I need to read up on that more, and I'm working my way through this paper, but I think there's evidence that there are some cues that predatory bacteria pick up on. And that's, as you said, like in direct contrast to what's going on. Because when they're out of a host cell just as particles, there's they don't have flagella, they're not bacterial cells, they're actually subcellular, so they're not going through these processes of chemotaxis. And so it's a very different way of thinking about search strategy when you're contrasting predatory bacteria with these bacteriophage. I think that's really interesting.
So I'm really glad that you talked about that. I actually, this is really just a side tangent to the paper, but I really, when I was doing research on writing a proposal, I read a lot about bacteria. I read a lot about E. coli and how chemotactic sensing of certain molecules can change the direction or change direction or movement in the flagella. And so it was really interesting. I actually have some books here if you're interested in reading them.
You just like,
totally side tangent. No. At any rate.
Any particularly good ones though? Did you of the books that you read?
I did find one, but I should probably talk to you. Oh, okay. Do you want to see?
I do.
Okay. So at least since we're talking about microbial motility,
we are. Or lack thereof. I think
there is this one, Chemotaxis, which is actually quite nice and very, very comprehensive.
That cover looks like it should be like a 70s cover band or something like that.
Oh yeah, it does, with like the green and the purple.
Now appearing in Chemotaxis.
So it's by Michael Eisenbach, and so there's a lot of different types of chemotaxis, and they talk about in here. So there's that one.
Okay.
And then I know I think there's one part in here, cellular motility. Actually, there's also this one, chemical communication among bacteria. This is edited by Bonnie Bassler.
Oh, okay.
So they have one chapter in here just specifically about motility, and I can't remember. There's like this special bacteria, and I can't remember if it's a bacterial or fungus. But at any rate, I can show that to you and tell you more about that later.
Yeah.
Okay.
No, and I think that's good because it sets up the challenge that these authors are dealing with. So when we think about bacterial cell motility, we think about kind of active processes of chemotaxis, we think about flagella, we think about different ways that bacteria have evolved to move actively through environments. And what this paper is discussing is this evidence that we're not going to have active motility with viruses. They're not going to be following a chemotactic gradient of any kind. But what they found is that it looks like in these mucus layers that there are particular interactions happening between molecules on the capsid on the outside of the virus particle and elements of the mucin is it the glycopeptides in the mucin?
Yes.
Yes.
So were you able to read the supplementary material?
I did not. No, I never did. I always end up saying I did not read the supplementary material, so all the answers are in there.
Well, no, there's two previous papers that they wrote before this particular paper, and the initial, because that one talks about how the hot proteins on the surface of the bacteria bind to the mucin glycans, and so that's I did read this a few years ago when it came out, and so my memory is a bit fuzzy on this, but
I'm impressed that you remember you read it. I can't remember.
Well, the reason why I read it is because the idea that phage, or that we have a commensal phage that protect us from our system, I thought that idea was really, really interesting, because we don't. There's not a lot of work that's going on right now about commensal viruses.
True.
Like, there's tons of work going on about our microbiome and commensal bacteria, but from all of the sequencing and everything that's been going on, like, yes, there's lots of microbiome research and it's great, but from my perspective as a PhD student, I was like, what's the next step of the research and where is it going to go? And so for me, I always thought that it's definitely going to go where we're going to talk about commensal viruses. And this whole discovery process is going to happen again, because now we have tools and the technology where we can now detect commensal viruses, and we have that were developed the biome viruses, but I mean, obviously they're much smaller.
Yeah, no, absolutely, though. I think that's very well said. Up to now, I think they've largely been ignored is unfair to the people who are studying the viruses, but overlooked, I'd say, by most microbiome studies, just I think for reasons of simplicity.
Yeah.
But you're absolutely right. I mean, you know, if we had a virologist here, they'd be able to quote the numbers to us about how vastly outnumbered we are by viruses. So you know, that's a significant part of understanding, in this case, understanding our immune response and understanding, you know, they suggest here that this is, you know, their last sentence here on the first page is you know, we propose the bacteriophage adherence to mucus model. This is their previous model. But they do say as a part of that, phages adhere to the mucosal surface of animals and provide a non-host derived layer of immunity. And that's a really neat concept. And I think you're absolutely right that this is where we need to go with understanding the interactions between microbes and ourselves, starting to incorporate viruses for sure.
It's just going to get more and more complex over time. It's like first we're like, oh, there's interactions between viruses, and now or with bacteria, and now there's interactions between viruses, bacteria, and then we're going to add in fungi, and then
yeah, I know. I was just going to say the fungal people are like, excuse me, let's talk about fungi, please.
or protozoans, like those things that live and eat your skin cells. And
absolutely. So, you know, bacteria are having their heyday right now, but I think they're going to have to start sharing the limelight. Rightfully so. Yeah, so no, I really like this, and I like that they included some. You can see in the paper part of the figures are illustrating exactly what they did technology-wise with this microfluidic chip. And I admit I am not a tissue culture person, so I imagine that this was probably very challenging to do, to try to replicate in a small chamber that's attached to a glass slide. Essentially, I think they're aiming for, in this case, it's lung epithelial cells. So they're trying to get a layer of lung epithelial cells started on this microfluidic chamber. And then they are trying to simulate the production of mucin and the flow rate that you would normally get in a human lung. That was what I gathered from it.
So if I could put this into perspective a little bit, because I work with tissue cultured cells on a regular basis, so then if you seed the cells onto any sort of plastic or glass covering, depending on the type of cells, they'll attach pretty easily, or they'll attach pretty easily, or they won't. But sometimes you need to coat the surface for the cells to attach. But if they have a system and they know that they have a set of tissue culture cells and that they'll secrete mucus, then it's not so bad. So like here in figure 1 in B and C, they show you like the growth of the cells over time, where they seeded the cells, and then they made sure to show you, okay, there's lots of cells that are actually in this microfluidic device. So all you need to do is if the cells will secrete mucus on its own, and you don't have to put anything where you've already adapted all the factors that it needs to secrete mucus. You just seed the cells on, and then they probably create some sort of flow using the that's what I would imagine, that they create some sort of flow using the microfluidic device for the mucus to flow over, which they can determine the flow rate. So it's actually quite elegant.
Yeah, and you can see certainly the fact that they've chosen lung epithelial cells. I mean, you can and they mention this, is you're certainly looking at things where mucus is a component of an illness. So like cystic fibrosis. I think that, you know, people who have cystic fibrosis are prone to getting lung infections, I think with maybe Pseudomonas and other bacterial pathogens. So certainly the application is immediately obvious in terms of trying to understand how phage might be able to play a role in providing an extra defense against a bacterial infection in these mucus-producing cells. So that's neat. Yeah, that's very neat. And it doesn't look like it takes up too much space in the lab, so also
Yeah,
convenient.
it was hard for me to visualize just from the figure like how big it was, but then when
Yeah.
they said that they put the chip on a single glass microscope slide, like single microglass microscope slide, they're about this big.
Yeah, yeah,
So
yeah.
it's actually pretty amazing.
No, I agree. I agree.
So one of their first finding is that they noticed that the number of phage and bacteria on the chip. So there's that the number of bacteria have decreased or are significantly lower than the number of than in the control group. So in their
Right.
in the T4 phage group, they say that because this T4 phage has the ability to bind to the bacteria with the Hock protein, that compared to the control or the T4 Hock mutant, that there's less bacteria. And so they show that there's not the T4 phage haven't, or there's less bacteria. So the implication is that the T4 phage have bound and lysed the bacteria. So did you think that was correct? What did you think about that result?
Well, I think my only question with this is really just more of an understanding question. So I understand their comparisons here are between wild type T4 and then they've knocked out these Hock proteins. And they're very plain about saying that their initial model that led them to knocking out the Hock proteins, as they went on here, they needed to refine it. So I think they do a very good job, and they're very clear about when they say like these were our initial expectations based on our previous data and our previous model, and then we actually had to tailor it a little bit. So they do a good job as they go on, and it's clear from their previous work that the Hock proteins are implicated in some way. And I think they originally thought that it was adherence and persistence in the mucus layer. But what they're finding with these data, especially in 1F, is that even the Hock mutants appear to be persisting in the mucus layer. So that doesn't look like that's what's going on with Hock binding, is it's not that it's allowing more phage to remain in the mucus layer, because they're not seeing a difference in the number of plaque-forming units with the wild type versus the Hock mutant. So then what they're seeing is a difference between the number of bacteria in the mucus layer, depending on if you have T4 or Hock. So they're seeing a greater reduction of the bacterial load in the mucus with the wild type versus the Hock mutant. And that prompts them to kind of investigate what's going on. The only question I had with that is the similarity in phage numbers. I was just trying to kind of think about logically, if you're starting with 10 to the 7th plaque forming units per mil, and then you end up with the same number of plaque forming units per mil in the T4 and the T4 Hock mutant, but you have I was trying to figure out numbers wise how that would work. So how are you, if you're not killing the same number of bacteria, how are you getting the same number of phage? That might be very obvious.
Because so you're saying that the killed bacteria, in theory, would release progeny phage,
right?
And so why hasn't the number increased in the T4 group compared to the mutant?
Yeah, I was curious about that, and maybe there's a really obvious explanation that I'm just missing because I don't consistently think about virus life cycles. But I was just kind of thinking, well, the way you get more phage is by producing phage in a hijacked host cell and then having that host cell release the phage. So that usually kills the host cell. And so I was just kind of curious why we've got the same numbers of phage, but we've got fewer bacteria in T4. I didn't really know.
So that actually brings up another question that I had, which is, why did, oh no, I might have just lost my train of thought. Because the phage, so, okay, you just said that the phage number doesn't increase, but phage number doesn't increase because, so they treated the mucin layers with the phage first.
Mm-hmm.
So they let the phage bind to the mucins, and then they added the bacteria to it.
Mm-hmm.
So then, would that be related? The fact that the phage might need time to adhere to the mucins. So if you had because they did that before. As far as I understand, it sounded like they did that before they added the flow, but maybe I could be wrong.
Okay, let's see. Oh, I see. I think I see what you're saying.
So if the phage haven't attached, and then in the new situation, the phage have lysed, you add the flow, but then before they're able to attach to the mucins, they've already flowed out of the system, so they don't contribute a significant increase.
okay,
That would be one thought.
Yeah, that could have something to do with it. I think it also, I can't recall with this experiment what the concentration of the mucin was, because I know that later on they find that that has an impact on the persistence of phage within that matrix, basically. Like they kind of call it a mesh. And the more concentrated it is, the smaller the pores in the mesh, and so the more entrapped the phage become. The lower the concentration, the more the phage can actually diffuse through the mucin layer. Yeah, maybe that has something to do with it. As I said, I think there's probably a very obvious explanation for why this is the case, but it's just something that I flagged because I was like, wait, I'm missing something here.
Or the other thing is that maybe it just doesn't. How much phage do you get out of one bacteria?
Oh, so that's depends on something called the burst size.
Okay.
The number of virus particles produced by any single host cell, and I think that changes based on virus. I think that's a factor that differs amongst different viruses. How many progeny they'll produce out of one infected cell. So I don't know for T4.
So is it possible that it wouldn't be detectable, like within that range, like if you have a slight increase? But
Could be. But I think I was curious because I know they said they perfused with 10 to the 7th per mil. Oh, maybe this is after all of that, maybe this is the source of my confusion, because I was looking at what they perfused with is 10 to the 7th per mil, but in their graph F, they don't actually say that it's a concentration. Maybe that's the actual absolute abundance. So that would make a difference. Yeah.
Oh, because it's like, but then it's the same question exists because. Oh no! So because you have,
I was thinking
that have the same number.
I was thinking that what they were showing was an actual increase. So I was thinking, because I had 10 to the 7th in my mind, and so what I was thinking is, well, on that graph, it should have started at 10 to the 7.
Oh yes, no.
And then it's jumped up to 10 to the 9 to 10 to the 10th. But maybe that's not true. So maybe that's just because it's a concentration, 10 to the 7th per mil. But in the graph, they might actually be showing total abundance. So they had however many mils, and so then they're getting 10 to the 9th, the 10 to the 10th is the total instead of the concentration. That could be why I'm getting confused.
Well, I think because I'm so used to because what I work on is I work on bacteria ligands that attach to host cell receptors, and so I was getting confused because I thought that there was actually a molecule on the phage that was binding to the bacteria, and that's what they were finding, but that's not it. They're finding that there's a molecule on the phage binding to the mucin. Okay, so that's different.
Yep.
So then that changes.
Yeah.
Because I was like, whoa, well, if it was able to bind to the bacteria and lyses the bacteria, if you don't have the protein, then you don't lyse the bacteria. That makes a lot of sense to me, but that's not what I'm saying.
Yeah.
Okay. All right. Okay, so within there, let's see, where do they go from here? So from here, they end up going from diffusion to phage. They move from showing us the system and then how many phage, and the fact that you have this decrease in bacteria from if you have the hot protein versus if you don't,
Right.
to having these two theories, which is you either have phage accumulating at a higher abundance, so you have more phage, or they persist longer in the mucus layer, so there's more of them in the mucus layer there for a longer time. And so to test this...
Yeah, so I think that was their previous model, right? So their previous model, they were hypothesizing that it was a numbers difference, that it was either abundance and persistence or something like that.
And so they found there wasn't that much of a difference.
Yeah, that's what F is showing them is, I think to their surprise, from what I could read, they were like, hey, okay, wait a minute, that's not actually what's going on. We're getting the same number of phage retained in the mucus layer. So clearly, when they show the difference in bacterial abundance, then they can start to say, all right, it's not the numbers of phage, but it's actually their efficiency at killing bacteria that there's some kind of a difference going on here.
So then from there, they use fluorescently labeled phage, and then on the microscope side, and then they detect and plot the patterns.
Yeah, the tracking. Boy, that was neat. High-speed multiple particle tracking. What did you do today? High-speed multiple particle tracking, you know.
Well, we use, because I'm used to tracking, or in a previous life, I worked on neurons, and we tracked vesicles in neuron axons, so using fluorescently tagged vesicle cargoes. So we would just watch them, but they were active. So but they would just go zoom this way, or zoom that way, or they would just go back and forth. So it's actually really neat to watch. But
it's like is it like a very
bigger than a virus?
Colorful tennis match. It's like a very colorful tennis match. Just like, huh?
Yeah, if you when you do the two color movies, like you just have it's like a highway. Just have green colored cars going one way and red colored cars going another.
That's very cool.
We're going back and forth, actually.
That's very cool.
But when they did this, they found that at a specific mucin concentration, you have a difference in the effect in the diffusion constant.
Yeah.
And so this is math that let's see, diffusion constant. This is math that I didn't there were some details I didn't particularly understand probably because I don't understand the math as much. But how did you interpret what they did?
Well, I liked that. So they were very clever to look at two different measures. So they've got two things in figure 2 in which they're trying to get at what's going on. So they know they've got phage in this mucus layer. Now what they're trying to figure out is how is this phage moving around in this mucus layer. So we know there's going to be random Brownian motion. And they make a point of saying, phages are inert particles dependent on random diffusion driven by Brownian motion to bring about their chance encounters with the bacterial host. So this gets back to what we were talking about at the beginning, which is that phage don't really have any active search strategies. They're just kind of, the collisions happen between phage and potential hosts just due to random motion. But they're thinking, okay, well, maybe there's something else going on with diffusion. So let's take a look at diffusion of these phage within a mucus layer. And they were really clever to look at two separate measures of this. So 2A is the effective diffusion constant, and then 2B is the diffusion exponent. And I did not look at the equations. I did not look at the supplementary material. But I think they did a pretty good job trying to kind of compare and contrast like what these two things measure different aspects of diffusion. And I think one of them is, and I want to look at it to make sure of this. So you've got diffusion. The diffusion constant is kind of, I think, has more to do with the distance, I want to say. And then and then the diffusion exponent, as far as I understood it, it's kind of, boy, I want to, let's see. Here's where I'll just say what they said. So the effective diffusion constant, which is what they're showing in 2A, and the diffusion exponent, which is what they're showing in 2B, are separate and independent phenomena in the sense that one cannot be taken as indicating the other. So the diffusion exponent kind of looks at basically the walk that phage particle takes in the mucus layer? And so rather than like having a measure of distance, like how far did it move, I think it's actually trying to capture more of the sense of what space is that phage particle moving in, rather than a total distance.
Well, so
that's how I interpreted it, although it could be totally wrong.
So based on how I understood it, which is what they did, they determined the diffusion constant based on the diffusion data that they were able to obtain, which I think you're totally right, is the amount of space or area that they're walking in per second.
Mm-hmm.
So if you have a higher diffusion constant, that means you walk more space in a given amount of time.
Mm-hmm.
So then for the T4 hawk, you get it walks more space per particular amount of time compared to the T4.
In relationship to the, in relationship to mucin concentration. So there's certain mucin concentrations where that's true, and then there's certain ones that is not true.
So then based on that, if I understand this correctly, they determine the diffusion exponent by plotting the data of the diffusion constant over time.
Mm-hmm.
Oh, has somebody joined us?
Yeah, hi Chris, how are you?
Hi.
I'm just trying to figure this out.
Okay, fair enough. Yeah, it is not the world's most user-friendly system, I have to say.
Okay.
Let us know if we can hear you and see you just fine. So if there's something that you need help with, let us know.
Sure.
Would you like to introduce yourself?
Sure. I'm Chris Upton. I'm I guess virologist / micro person doing bioinformatics and occasionally interested in phage. So I'm at the University of Victoria in British Columbia.
Great. Well, thanks for joining us.
Okay.
I'm in Vancouver at SFU. I spent a summer with Dr. Francisciano at UVic, maybe about 3 years
just
ago.
down the hall.
Yes, but we're really happy to have you, especially because you're a virologist, so you can bring a different type of perspective to this journal club.
I've got to try and figure out how this software. Twitter was so much easier.
Oh, I know. Yeah, yeah.
So, in terms of your perspectives on the paper, what did you enjoy about it, or what did you think about this particular paper that we've been reading?
You're asking me?
Yes.
Paper.
You just get put right on the spot when you.
I haven't actually read the paper. I was just going to lurk and read the Twitter comments.
Oh, sure. No, that's fine.
In my lab that's come up from Brazil for 6 months, and he's kind of interested in phage and composting. And I just thought, oh, that's kind of cool. I'd just sort of see what was happening.
Yeah. Yeah, so we were just kind of getting at, so yes, absolutely, feel free to lurk, and since you're a virologist, you might catch us if we say something that's wrong, so feel free to jump in if we're way off.
Sorry. I just, because somebody joined us, I wanted to feel like you were involved and welcome. But yes, you can feel free to lurk as well.
Yeah, so we were just looking at this. They've got this microfluidic chamber that they've used to try to replicate mucin-forming epithelial cells, and I'm really impressed by this because I kind of think that what they're teasing out here is not the easiest thing to tease out. So they have T4 phage, and then they have T4 phage where they've knocked out the Hock genes, so they don't produce this protein on the outside of the virus capsid. And they thought that that would have to do with binding to the mucus layer. So they kind of expected that when they got rid of the Hock proteins, that meant that there would be fewer phage persisting in the mucus layer. And they found that that's not actually the case. And what they're looking at now is how these T4 phage are actually encountering potential bacterial host cells in the mucus layer and what the role of the Hock proteins are. So we were just looking at how they calculated diffusion in the mucus layer, which I think was a pretty clever effort as far as I can tell. I thought it was clever. And so they've looked at the diffusion constant, which Karen was just talking about, and then also something called the diffusion exponent, which really, Karen, sorry?
All right, no, I'll let you finish, I'm sorry.
No, that's okay. I was just going to say that, and I think they were wise to look at both of these things, and they looked at them across a concentration, different concentrations of the mucin. I think if they'd not done that, they might have missed this completely. So I think it was well constructed to be able to try to tease this apart. I think they used that system, that chip that they developed. I think they used that very well.
Right. So I was just going to mention that I think the way that they came up with the exponent is by plotting the curve of plotting the curve of the fusion constants over time, I think, and then by looking at so then that exponent is an indication of the relationship of that curve. So if you have a curve that equals 1, your curve looks like a certain way. I, again, did not read the supplementary method, but I'm sure that if I look at them that the graphs will look different. And so that relationship, with you have superfusion, which you have, your curve is going to look a certain direction, and then if you have subdiffusion, your curve is going to be less than 1, which looks different.
Right. So that, all their data in Figure 2 combined basically points them to the fact that wild type T4 in certain mucin concentrations looks to be demonstrating subdiffusion. So it's not just going to be, it doesn't look like what they expect for random Brownian motion. They're actually detecting something that looks like subdiffusion instead of, if it was random, it would be along that dashed line, but they're seeing it dip below that for certain concentrations of mucin. So that, I think that was their first indication that there's something going on in terms of how the phage are interacting with mucin, and that the Hock proteins are involved.
And so when they talked about subdiffusive motion, like for the longest time, I was just like, what does subdiffusive motion look like? And so that's why I found, and I didn't realize that what it really meant is they show that in Figure 6. So here you have this, here they have the Brownian motion trajectory, which kind of looks like this. So it moves randomly back and forth in wide spaces. But then for subdiffusive motion, here they're saying it's more, it doesn't move as much away from the original starting point. So it's kind of like I'm going a little bit here, and then so it's more, I don't know if I would be describing this correctly. So it's more like this, or what do you think, Laura?
No, I think you're right. I think I think they're making a contrast between the way it would regularly move just by random chance, and then when it's in the mucin mesh, there's an interaction that's happening that gives it a different type of motion. And I think you're right that I did read, they said that this subdiffusive motion is going to restrict the phage to a smaller search space than it would otherwise have by expectation for a random motion.
Right. So, I mean, when they say that, like from my interpretation, it just means that the phage is not moving too far from the location where it's actually located. Whereas in Brownian, it would go like this. But it's really weird that that's not active. Like that they say when you think of hunting strategy, you think of something that the hunter is actively doing. But like you mentioned before, it's not, the phage is not doing this actively.
Nope.
So how does it, and it's doing this through some sort of interaction with the mucin because, or wait, no, did we just disprove that the binding is not, is not responsible, or?
No, I think it is. I think the otherwise you'd get the Hock protein. That's why knocking out the Hock proteins gives you random Brownian motion across all those concentrations, and you see subdiffusive when you've got those Hock proteins. So it looks like what's happening is, and I'm trying to think if they did. No, what I did not read in here was, and maybe they've done this before, is looking at like the binding affinity between HOC and the mucin glycoproteins.
Right.
I don't know. They might have already done that. It's not in here, but I didn't think I read anything in here about that. So what they're suggesting is that it looks like there's transient interactions between HOC on the outside of the phage and mucin. So it's almost like, I was trying to think of a good analogy for it, and I just couldn't come up with one. It's almost like if you did the Price is Right game, Plinko, where you kind of let the little disc go and it bounces down amongst all those different pegs. It's almost as if the peg held it for longer, right? You know, that's the way I'm visualizing it. It's like it holds it, and then it lets it go, and then it holds it, and then it lets it go, and then rather than just ding, 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 ding all the way down.
That's a really, really good point. I think that's very cool, because the fact that they would have transient interactions or transient affinities with the mucin, so then it kind of is like sticky for a little while rather than being able to move back, or rather than moving back and forth like this.
But that's a stupid question. Hang on, let me just close my door. So, what mucosal surfaces are they referring to, or do they think this is happening at?
In this experiment, they have an in vitro system where they've got that chip. But I think they're...
Right?
Sorry?
So they are looking at like a model of an interaction.
Yeah, so they're using lung epithelial cells. So I think what they're hoping to do is try to, at some level, model what's going on in the lung. But they do make a point at the end of it that that's a far more complex environment than they're able to capture with just this model, which is true.
So I guess my question is, always as an unbeliever, is as soon as the phage, so the phage is going on and off, so it's held against the mucosal membrane. I can believe that, but as soon as it gets away far enough that it is unlikely to come back, then that interaction will never happen again until it comes back to a surface. Unless it's happening in, I don't know, are they saying it's happening in normal, like just fluid in the lung?
Yeah.
What is the surface? Is it strictly against the cell, or is it interacting with sort of goop that's just sort of...
Yeah, I think it's as near as I can understand it, they've got the layer of epithelial cells, and then what they're looking at is the kind of mucus mesh or the mucus matrix that gets secreted from those epithelial cells and is constantly kind of building up, and then they have a flow going across the top of it that's sloughing off the top layer.
Okay.
So it's kind of getting exuded, and that's the mucus layer is what they're looking at specifically.
It's kind of a 3D layer almost. It's not just
exactly.
A flat surface.
Because they do, in their experiments, they do vary the concentration of mucin, and I don't know how they did that, but they do have up to like 4%. So they're trying to kind of do it as a denser mesh and as a looser mesh, so they get different pore sizes for the phage particles to make it through. Mm-hmm. And they really only see this subdiffusive motion at a certain range of those concentrations. And that's an effect, I think, of what you were talking about, is how frequently are the particles going to interact with the mucus mesh or the mucin.
I mean, in a body or in a tissue, you know, how much is taking place at a surface and how much in sort of a lumen, sort of, where it never comes in contact with the surface?
Yeah, that's a good question. I think that would be something that they would probably want to try to address if they developed the model farther.
Well, and I think they mentioned that to a certain extent, where they talk about I guess they don't talk so much about the surface of the epithelia, but they do talk about how the thickness of the layer also will increase or decrease the probability of encountering a bacteria. Or the concentration of bacteria. So if you have a lot of bacteria, the phage are going to be able to encounter bacteria more often, so then the subdiffusive motion isn't as relevant in those cases. But what is interesting is that when we talk about like how much will this mimic the in vivo situation, because it's at such a kind of like a limited optimal concentration range, is how much would subdiffusive motion actually happen in real life is a very interesting question.
Yeah, that's true. I don't have a good sense for that physiologically. Yeah.
Well, because well, I don't. Yeah, so like because you don't never know how much phage there are targeted towards specific bacteria, or because there's one for the phage to find its target bacteria, it's going to take, if you have more bacteria, it's going to take much longer than if you have monopopulations of the exact phage that targets the exact bacteria.
Oh, so what you're saying is when you increase the complexity of the system by adding
Yes.
different types of bacteria,
Yeah.
unless you have a phage with an incredibly broad host range, you're going to have an extra obstacle to encounters. So not only is the phage dealing with the mucin layer, then it's dealing with the obstacles of having other bacteria that it's not able to attack. I see. Yeah, that's a good point.
Well, because the authors are, well, there's a general interesting question, which is the authors are saying that this is a very good way of selecting for phage in phage therapy. So they're saying if you select for phage that have optimal subdiffusive motion strategies, then that type of phage might be a better choice for a phage therapy, for a type of phage therapy, or for phage therapy, or it might target the bacteria, or whatever the phage therapy target is better. So I guess I would ask, in your opinion, do you think that that's true? Would selecting for subdiffusive phage be a beneficial trait in phage therapy?
I don't know. I think that's a good question. I think if you're going to deploy phage as a possible treatment against bacterial infection, like let's use the lungs as an example. So you have a cystic fibrosis patient, and you want to deploy phage as a way of combating bacterial infection in the lung. What I don't know about this system is how well it addresses the mucus layer in a cystic fibrosis patient, for instance. So if you've got somebody who's ill with cystic fibrosis, their mucus layer is going to have its own particular characteristics compared to a healthy person. So you're going to be looking at how does it deal with moving through a specific phage, how is it going to deal with moving through the mucus layer of a patient with cystic fibrosis, and then is it going to be able to attack, for instance, Pseudomonas? Is it going to be able to find and attack those cells? Is subdiffusive motion the key factor there? I'm not sure.
Well, that brings up another really good point, which is, would cystic fibrosis patients have less bacteria in their mucins compared to normal microbiome? Because if you have less bacteria, then subdiffusive motion comes into play more, rather than if you had more. Or maybe it's just diversity, but I guess just the sense of in vivo ratios is important here. But if there is a disease condition where you have an in vivo ratio where it's like, okay, you want to get rid of these bacteria that are colonizing the mucus layer, and there's not a lot of other type of bacteria that are present there as well, maybe this is an effective strategy because there's nothing else preventing the phage from targeting its target.
And it seems like in cystic fibrosis, most people are trying to get rid of the mucus as fast as possible. That's the problem. So if your phages actually are getting stuck in the mucus more than another one, it might be. I'm not sure. It seems like it's a big advantage. I don't know. I just kind of interested that it hangs around in mucus, but it's like.
So for cystic fibrosis, they produce more mucus than normal.
Right, and they're trying to. I mean, half the treatment is what therapy is, trying to get rid of that stuff, right? I don't know where the infections come in and where they cause the most problem in cystic fibrosis.
Yeah, I'm not sure on that either. For some reason, I've got it at the top of my head, basically some association between Pseudomonas and cystic fibrosis.
Right. I don't know if it's chronic infections or infections or
I'm not sure.
infections, twice a year type thing, which would be bad for people, or if it's always there.
Yeah.
So we're coming on about the end of our time here. I'm just wondering, does anyone have any final last thoughts that they'd like to add about the paper? Or
I should have read the paper. I only got as far as the abstract. Well, I was trying to
No,
at the same time.
honestly though, this is very informal, and we're always happy to have people hop in and provide some perspective. So I do want to thank you for joining us. So we really appreciate that.
That was fun.
No, I would say I'm really glad we read this. It popped up across my Twitter feed, and as I was telling Karen, it's one of those papers where I saw it and I thought, I'd really like to read that, and there was no way I was going to do it unless I had to show up for this and talk about it. So I'm glad we did this paper. I think it'll be really interesting to follow this. It's clear that this is a persistent line of work for these groups. They mentioned in the paper that this is a refinement of a model that they have been working on. And so I'll be interested to see where they take this, especially with some of the things that we've just been talking about in terms of practical applications to more realistic situations. So they've got this good in vitro system to help them tease apart things like how does the mucin concentration affect what we observe with subdiffusive motion. But now what they can do is they can try to maybe introduce some more complexity, try to see if they can replicate some of the things you'd see in cystic fibrosis, and figure out if we're going to use this as a way to understand phage therapy, what more do we need to know before we try to kind of do this in an in vivo system.
yeah, I think that's a really good note to end on. And so thank you, Chris, for joining us, and well, thank you for everyone else for watching and joining us on air. And so for those, this will be live on YouTube, and yes, so if you want to join us again next time, we'll have it about once a month, hopefully. And so we'll hope to, yes, Chris, is there something you wanted to say?
Yeah, I was going to say, is there a email notification system for when these?
There is not, but there can be. I haven't really hit upon the best way to keep people in the loop on these. Email would be, I'd be happy to do that if that's more convenient for people.
Something like, or an RSS feed on the web page or something like that.
Yeah,
Yes,
so we do
there
update
is.
the web page, and we have been tweeting, but I think one of the problems with that is that if you're not online when that tweet goes out, then you just don't see it.
That's right.
So I shied away from email because I know everybody's inbox is already really full, but if that's a better format, I'm happy to do that.
So I think we can also have submission to the RSS feed, or maybe there's a way for us to create a mailing list or some sort of informative thing through WordPress, which might actually be possible. But we will look into that. Okay.
Yes.
All right.
Yeah.
Well, thank you everyone for joining us. And so
absolutely.
this is us signing off. Bye.